The microscope has a fascinating history that spans centuries, revolutionizing our understanding of the world. From its early beginnings to modern innovations, the story of the microscope is one of curiosity and scientific breakthroughs. The concept of magnification dates back thousands of years. Ancient Egyptians and Mesopotamians used polished crystals to magnify objects, while the Romans discovered that glass could make things appear larger. However, it wasn't until the late 16th and early 17th centuries that magnification's true potential began to be realized. In the 1590s, Dutch spectacle makers Hans and Zacharias Janssen are often credited with inventing the first microscope. Their device, consisting of two lenses in a tube, could magnify objects up to 9 times. Though impressive, it was far from the microscopes we know today. Galileo Galilei, the famous Italian scientist. In 1609, Galileo heard about the Dutch invention and decided to make his own version. He created the occhiolino, or little eye, which could magnify objects up to 30 times. While Galileo is more known for his contributions to astronomy, his work with magnification paved the way for future developments in microscopy. However, it was Anton van Leeuwenhoek, a Dutch cloth merchant, who revolutionized the field of microscopy. Born in Delft in 1632, Leeuwenhoek's work with magnifying glasses to inspect the quality of fabric ignited a lifelong fascination with lenses. Unlike his contemporaries who used compound microscopes with multiple lenses, Leeuwenhoek crafted simple microscopes with a single high-quality lens, some as small as a pinhead. These microscopes could magnify objects up to 270 times. What set Leeuwenhoek apart wasn't just his lens-making skills, but his insatiable curiosity. He observed everything he could: pond water, insects, blood, and even the plaque from his own teeth. In 1676, he made a groundbreaking discovery: tiny organisms moving in a drop of water, which he called animalcules. This was the first recorded observation of microorganisms, a discovery that would forever change biology. Initially, Leeuwenhoek's findings were met with skepticism. The notion that tiny, invisible creatures lived among us seemed ridiculous to many. But as he refined his techniques and shared his findings with the Royal Society in London, the scientific community began to accept his work. In 1683, Leeuwenhoek became the first person to describe bacteria, observing little animalcules in the plaque from his teeth. His discovery of bacteria was a monumental breakthrough, reshaping our understanding of disease and biology. While Leeuwenhoek was uncovering these microscopic worlds, Robert Hooke, an English scientist, was improving the design of microscopes. In 1665, he published Micrographia, a book filled with detailed illustrations of objects as seen through a microscope. Hooke coined the term cell in this work, describing the basic building block of all living organisms. His microscope, more complex than Leeuwenhoek's, used multiple lenses to achieve higher magnification. However, it suffered from chromatic aberration, a distortion caused by different wavelengths of light being focused at different points. This issue would plague microscope design for decades. The 18th and 19th centuries saw significant improvements in microscope technology. Achromatic lenses, developed in the 1730s by Chester Moore Hall and later improved by John Dolland, corrected chromatic aberration, allowing for sharper images at higher magnifications. In 1830, Joseph Jackson Lister made another breakthrough. He developed a method for combining lenses that greatly reduce spherical aberration, another common issue in early microscopes. This advancement led to the creation of the modern compound microscope, paving the way for more accurate and detailed observations. As microscopes improved, so did our understanding of biology. The development of cell theory by Matthias Schleiden and Theodor Schwann in the 1830s was a pivotal moment in science. Their theory, which states that all living things are made of cells, became one of the fundamental principles of biology, influencing research for generations to come. The 20th century brought even more exciting advancements in microscopy. In 1931, Ernst Ruska and Max Knoll invented the electron microscope, which used a beam of electrons instead of light to create an image. This allowed for much higher magnifications and the ability to observe structures as small as individual atoms. The electron microscope opened entirely new fields of study, such as virology, allowing scientists to observe viruses for the first time. These discoveries played a critical role in developing vaccines and treatments for diseases. The invention of the electron microscope wasn't the end of microscopy innovations. In 1981, Gerd Binnig and Heinrich Rohrer developed the scanning tunneling microscope, which allowed scientists to create three-dimensional images of surfaces at the atomic level. This breakthrough was crucial in advancing nanotechnology, enabling researchers to manipulate matter at the atomic scale. Around the same time, the confocal microscope was becoming a game-changer in biological research. Although it was invented by Marvin Minsky in 1957, it didn't gain widespread use until the 1980s. The confocal microscope allows researchers to generate detailed 3D images of biological samples, revolutionizing fields such as neuroscience and developmental biology. As we moved into the 21st century, super-resolution microscopy techniques pushed the boundaries even further. These techniques, developed in the early 2000s, allowed scientists to observe structures smaller than the diffraction limit of light, a barrier that had previously limited the resolution of optical microscopes. One of the most significant techniques was stimulated emission depletion, STED microscopy, developed by Stefan Hell in 1994. STED uses laser beams to achieve incredibly precise imaging with resolutions as low as 20 nanometers. Another groundbreaking technique was photoactivated localization microscopy, PALM, developed by Eric Betzig and his colleagues in 2006. PALM works by activating and imaging a small subset of fluorescent molecules, allowing researchers to precisely locate individual molecules within cells. These super-resolution techniques have enabled scientists to make groundbreaking discoveries, such as studying the intricate structures of synapses in the brain and tracking the movement of proteins within living cells. The impact of microscopy on medicine, biology, and material science is immense. In medicine, microscopes have been crucial in understanding disease causes and developing treatments. Observing bacteria and viruses has led to antibiotics, vaccines, and advanced diagnostic tools. In pathology, microscopes are used daily to diagnose diseases like cancer, allowing for earlier and more accurate diagnoses. In material science, electron microscopes have allowed researchers to study materials at the atomic level, leading to stronger alloys, more efficient solar cells, and advancements in nanotechnology. Scientists can now manipulate individual atoms, opening new possibilities for creating materials with unprecedented properties. As we look to the future, the potential for microscopy is boundless. Researchers are developing new techniques, such as combining microscopy with artificial intelligence and machine learning. AI-powered microscopy can automate the process of analyzing images, leading to faster and more accurate discoveries, particularly in fields like drug discovery. Another exciting development is light sheet microscopy, which allows for rapid 3D imaging of large biological samples with minimal damage. This technique is proving invaluable in developmental biology, where researchers can observe entire embryos in real time. The story of the microscope is far from over. From its humble beginnings with simple lenses to today's super-resolution techniques, this remarkable tool has continually pushed the boundaries of our understanding. The future promises even more incredible discoveries as microscopy continues to shape how we perceive the microscopic world and the universe itself.